So, in the line of protein synthesis, your next organelle है that is lysosomes. Lysosomes, they are one structure that buds off from Golgi complex containing the proteins. अगर डिस्कवरी के हवाले से बात करें then these were for the first time seen by D. Douve, and these are found mostly in animal cells because they are digestive organelles. They are mainly involved in digestion. अब digestion किसकी होती है? It can be anything. It can be the cellular organelle. आगे चल के उसकी मैं डिटेल में बात करूंगा, but their primary function is digestion. And if we look at the composition, enzyme composition of any lysosome, there are at least 50 different hydrolytic enzymes. जी कोई problem? There are at least 50 different types of hydrolytic enzymes which are produced by rough endoplasmic reticulum, and they are processed, and then they are packed in this lysosome. These enzymes can virtually digest any biomolecule, any macromolecule. अब macromolecules की अगर definition करें, you can name any carbohydrate, protein, lipid, or nucleic acids. These are categorized as macromolecules because they are polymer of the subunits. जो एक प्रॉपर्टी स्पेसिफिक प्रॉपर्टी है लाइसोसोम्स की बिकॉज दे आर फाउंड इन साइटोप्लाज्म. और अगर हम पीएच की बात करें, साइटोप्लाज्म की पीएच न्यूट्रल होती है, बट लाइसोसोम्स की जो है लाइसोसोमल पीएच जो है दैट इज मोर एसिडिक. Therefore, we have lower pH values inside the lysosome, and this acidic pH is basically required for digestion process. You might have observed कि विद इन लैब अगर आप एसिड ट्रीटमेंट करें किसी भी मैक्रोमोलिक्यूल की, लाइक शुगर को एसिड से ट्रीट कर लें, या फिर आप ऑयल को, या फिर आप कार्बोहाइड्रेट्स को, किसी को भी एसिड ट्रीटमेंट दें, then there is possibility of quick digestion. So acidic environment is present within the lysosomes, and the pH which is present in the lysosome is 4.6. Now the question is how this pH is maintained. This pH is maintained because of the intralysosomal high protons, and this proton concentration is maintained by one proton pump that pushes the protons within the lysosome at the expense of energy which is derived from ATP. इस अगर पिक्चर को देखें, दिस बाउंड्री टेल्स अस द लाइसोसोमल मेम्ब्रेन, और इसके अंदर जो एंजाइम्स हैं, प्रोटीएजेस, ग्लाइकोसाइडेजेस, न्यूक्लियेजेस एंड लाइपेजेस, they target their substrate, and the substrates are digested into the simpler compounds, and then these digested compounds, they are assimilated in the cytoplasm, which can later be used by the cell for multiple activities. So the digested component is released in the cytoplasm for cellular usage. We can also claim that the lysosomes are full of a bag full of hydrolytic enzymes, a bag full of destructive enzymes, and their primary task is to do the digestion. अगर हम देखें इसका फंक्शन ह्यूमन सेल्स या यूकैरियोटिक सेल्स से हट के, then even they are involved in digestion of food particles in certain phagocytic cells in microorganisms, like which are deprived of any specific digestive system. Their cytoplasm have the lysosome, and the lysosome perform the digestive function over there. In human body अगर हम immunity को देखें, immune system को देखें, या blood system को देखें, not immune system, blood system को देखें, तो lysosomes, they have important significant role in phagocytic activity of defense cells. Of defense cells में, white blood cells are of multiple types. Neutrophils, these are the phagocytic cells or defense cells. Similarly, लिवर में कुछ सेल्स ऐसे पाए जाते हैं जिनकी फेगोसिटिक एक्टिविटी होती है. दे आर कॉल्ड एज मैक्रोफेजेस बिकॉज ऑफ देयर साइज. Within these phagocytic cells, lysosomes are very much active. जब हम बात करते हैं माइक्रोबियल इन्वेजन की, और माइक्रोबियल इन्वेजन मींस बैक्टीरियल इन्फेक्शन की, एंड इफ द बैक्टीरिया आर phagocytosed by the cell, these are taken up by the lysosome, and their activity is, the bacterial activity is diminished because of the reason, or is like halted because of the acidic environment of the lysosome, and subsequently the digestion of that bacterial cell starts due to the action of these enzymes. एंजाइम्स को अगर हम देखें, कुछ एंजाइम्स का नाम अगर हम देखें, देन देर आर फॉस्फेटेजेस एंजाइम की क्लास. एसिड फॉस्फेटेजेस which target the phosphomonoesterases, then acid phosphodiesterases which target the diesterases. Enzymes में nucleases को देखें, then there are some enzymes which target the RNA, so acid ribonuclease, acid deoxyribonuclease which target the DNA. Ribonuclease कहीं पे भी आप बात करेंगे, they are targeting the RNA. Deoxyribonuclease will target the DNA, and probably these are called as acid enzymes because they are working in the acidic environment. For proteins, we have cathepsin enzyme, and for collagen digestion, we have specific enzyme which is collagenase. Then for carbohydrate and oligosaccharide polysaccharides के लिए fucosidases, fuco fucosyl oligosaccharides, and for manosidase is for manosyl oligosaccharidase, means for mannoses these enzymes are present. इसी तरह से आप कोई और भी नाम कार्बोहाइड्रेट का सोच रहे हो और उसका भी एंजाइम स्पेसिफिकली एग्जिस्ट करता है, जैसे हमने कल गोल्जी कॉम्प्लेक्स के हवाले से बात की थी. ये जो पिक्चर है, दैट पिक्चर इज a modified, a different version of the lysosomal activity. If you can look at the color scheme of this picture, the lysosomes are stained darkly blue, and the Golgi complex and sorry, the Golgi complex is pink. Lysosomes are also pink, containing the enzymes, and then there comes the food vacuole, and there is fusion. Then you can see that the digestive vacuole is changing its pH, and digestion is ongoing. Once digestion is completed, the undigested remains are either expelled or out of the cell, or they may remain within the cell. अभी उसके बारे में बात करते हैं. This is one electron micrograph जो कि मैंने आपको ऑटोफेगी के पॉइंट ऑफ व्यू से दिखानी थी. अगर हम लाइसोसोम का किसी भी एंगल से जो है वो देखें, फंक्शन देखें, फेगोसाइटोसिस इज द प्राइमरी फंक्शन, और उसमें अगर सेलुलर लेवल के ऊपर देखा जाए, देन ऑटोफेगी इज परफॉर्म्ड बाय द सेल दैट नीड्स द लाइसोसोम्स. अब यहाँ पे ऑटोफेगी और ऑटोफेजी का मतलब क्या है? दी सेलुलर टर्नओवर. इट्स नॉट ऑलवेज कि एक सेलुलर ऑर्गेनेली अगर मौजूद है तो वो एवर लास्टिंग है. इट विल बी टर्नड ओवर, इट विल बी रिसाइकल्ड, और उसके लिए जो है सेलुलर ऑर्गेनेली लाइसोसोम प्लेज इट्स रोल. When lysosome takes up the cellular organelle, it makes a structure जिसको हम autophagosome का नाम देते हैं. Autophagosome, this is the mitochondria which is taken over, or which is fused by the lysosome, and now this is going to be autophaged, or digestion हो जाएगी इसकी, और again the same thing, the digestive remains are going to enter into the cytoplasm. These uh, autophagosomes, when we consider that autophagy is a normal process, or this happens under some specific condition, experimental studies have shown that if you leave the cells, or if you culture the cells under deprived nutrient deprived condition, or you make those cells to starve for quite much time, then cells start doing or exhibiting this behavior of autophagy, and the lysosome will start eating the cellular organelles to generate energy. Cannibalism आपने सुना होगा, in which human, it is said that human eat human flesh, but this is not like that. When you start eating your own parts, then that is also referred as cannibalizing or cannibalism. So when a cell has its organelle, this one lysosome is is going to eat its another organelle, then this can be referred as a cannibalizing behavior of the cell. The fate of this autolysosome is that after digestion is completed, then the next vacuole which has undigested remains, that is referred as residual body. Now, this residual body has two fates. Either it goes out of the cell, the undigested remains are expelled out in the form of exocyte in the by the process of exocytosis, or it can remain within the cytoplasm for longer period of time, and in that condition this is now called as lipofuscin pigmented granule. And this lipofuscin granule basically is also another indicator of aging, process of aging. यहाँ पे एक कॉमन से एग्जांपल दी जाती है कि न्यूबॉर्न बेबीज आर हैविंग फेयर स्किन, but as the age grows, the age increases, and it goes leads towards the old age, the skin tone is going to get darker and darker. One of the factor of this change in tone is these lipofuscins, because when any pigmented material enters into the cell, and it can be anything which can enter into the cell, the pigments, they are not digested. Rest of every biomolecule is digested, pigment is not going to be digested, so that pigment will stay within the cell. Ultimately change in the pigmentation of skin will occur. Further, what molecular pathways are included in autophagy? uh, we are not going to highlight any specific pathway. I will just only mention here that there are certain specific genes, ATG, that is derived from autophagy. These AT genes, they are required to perform the process of autophagy, और autophagy pathway को जो है वो induce करने के लिए इन genes का expression ज़रूरी है. So the point to tell you here is every process, every action needs a protein message, and that comes from the genes. जैसा कि हमने पहले भी बात की थी कि सेल में जो भी फंक्शन होगा, that needs a message, and the message either will come from out of the cell, or it will be generated within the cell. And this message is always in the form of a protein. And for autophagy, अगर आपसे पूछा जाता है कि what type of genes are involved, so these are AT genes which are responsible for autophagy process. यहाँ तक बात क्लियर है जी?
सर एक क्वेश्चन है,
हम्म.
हम इन्हें डाइजेस्टिव ऑर्गेनेलिस कह रहे हैं, डाइजेशन मेनली कर रही हैं, डिस्ट्रक्शन कर रही हैं, तो ये सारे फंक्शंस तो प्लांट्स में भी होते हैं, फिर ये मोस्टली प्लांट्स में क्यों नहीं होती?
प्लांट्स में होते हैं फंक्शन, प्लांट्स में क्यों नहीं होती, इसका जवाब मैं आपको ये दूंगा कि प्लांट्स में इस लिहाज से जो है वो इस लेवल तक फंक्शंस नहीं होते कि वहाँ पे लाइसोसोम्स की ज़रूरत पेश आए. फॉर द रीज़न कि प्लाज्मा मेम्ब्रेन को अटैक करना इज़ वेरी इजी एज कंपेयर टू सेल वॉल. दिस इज़ रिलेटिवली इम्पॉसिबल कि कोई बैक्टीरियल सेल जो है वो प्लांट सेल के अंदर एंटर हो बाय द प्रोसेस ऑफ ऑटोफेजी. बिकॉज आप ये भी देखें कि एंडोसाइटोसिस जो है इज इट विजिबल इन प्लांट सेल्स? द आंसर विल बी नो, बिकॉज दे हैव देयर आउटरमोस्ट कवरिंग एज द सेल वॉल. नंबर वन. नंबर टू, लाइसोसोम के पैरेलल जो है कुछ और ऑर्गेनेलिज भी मौजूद हैं विच परफॉर्म सम स्पेसिफिक फंक्शन. अभी नेक्स्ट उसके बारे में बात करेंगे, देन यू विल गेट दिस आंसर।
सर एक और क्वेश्चन भी था।
हम्म।
सर आपने बोला है कि पिगमेंट्स जो है ना वो डाइजेस्ट नहीं होते, तो इस
जी
वजह से स्किन स्किन कलर टोन जो है वो डार्कर होता जाता है।
जी जी।
लेकिन सर वाइटनिंग क्रीम्स होती हैं, वो ऐसा क्या करती हैं कि वो स्किन कलर एकदम से चेंज हो जाता है, वो पिगमेंट्स के साथ ऐसा क्या करती हैं?
देखिए, दो इसमें थ्योरीज हो सकती हैं। एक तो जो है आप कह लीजिए कि इफ़ यू आर यूजिंग एन एंजाइम बेस्ड क्रीम, तो वो एंजाइमेटिक रिएक्शन अपना शो कर सकती है, और जो पिगमेंट है, डेफिनेटली पिगमेंट हैज़ इट्स ओन केमिकल कॉम्पोजिशन, तो उसको जो है वो ब्रेक डाउन करके करके या उसका जो कलर है वो फेड किया जा सकता है। जैसे ब्लीचिंग जो पावर होती है, दे दैट पावर इज रिस्पॉन्सिबल टू मेक द डार्कर कलर्स टूवर्ड्स द लाइटर टोन। लेकिन ब्लीच के इफ़ेक्ट्स जो हैं वो काफ़ी स्ट्रॉन्ग होते हैं, दैट कॉजेज द major damage, that may cause major damage to other cellular components as well, नंबर वन। नंबर टू, जो एंजाइम बेस्ड नहीं है तो उसके बारे में तो फिर यही है कि देयर केमिकल्स बेसिकली टारगेट द सेलुलर पिगमेंटेशन, और उसको जो है वो टोन चेंज करते हैं। जिस कंटेक्स्ट में आप बात कर रही हैं, इसमें जो है हमें जो नेचुरल जो है पिगमेंट ऑफ द स्किन विच इज़ मेलानिन, बेसिकली या तो हम उस मेलानिन को जो है वो प्रोसेस को इनहिबिट करते हैं ताकि मेलानिन इतनी ज़्यादा प्रोड्यूस ना हो, टू गिव द टोन और डार्क टोन टू द स्किन, or we interfere or we remove that melanin from the cell, so that using these chemical creams, so that the melanin is not there, and the tone will become lighter and lighter.
ओके सर, थैंक यू।
But keep in mind, chemical treatment with the cells is always dangerous, because you never know which chemical is going to interfere which cellular communication pathway, and turning it towards some disaster. Okay, then comes the lysosomal diseases. Apparently अगर देखा जाए तो लाइसोसोम बहुत माइनर सी जो है ऑर्गेनेली के तौर पे विजिबल होते हैं, but in case if there is any mutation, there is any problem with any of the enzyme which is a component of the lysosome, then it may lead towards a serious disaster. If you look at this picture, आपको ये enlarged vacuoles visible हैं और multiple vacuoles visible हैं. These are basically enlarged lysosomes. अब होता क्या है कि एक एंजाइम है, उसका जो है वो जीन म्यूटेट हो गया, या उसकी प्रॉब्लम हो गई, एनी काइंड ऑफ प्रॉब्लम दैट इट इज प्रेजेंट इन द सेल बट इट इज नॉट पैक्ड इन द लाइसोसोम, तो उस एंजाइम का जो सबस्ट्रेट है दैट इज गोइंग टू गो इन द लाइसोसोम, और वहाँ पे चूंकि उसकी डाइजेशन नहीं हो रही, तो अल्टीमेटली उसकी वो जो है वो स्टोरेज साइट बनती जा रही है। Lysosomes become storage site of those biomolecules. Ultimately, those lysosomes will start increasing in size, and there will be multiple enlarged vacuoles. एक स्टेज ऐसी भी आ सकती है कि एक सेल का कंप्लीट लाइसोसोम जो है वो इन कंप्लीट साइटोप्लाज्म जो है वो इन लाइसोसोम से कवर्ड हो, and cell will have everything pushed aside. So these kind of diseases are referred as lysosomal storage diseases or storage disorders, because their enzymes are non-functional, and the substrate is going to accumulate in the lysosome, and it is going to create trouble for the cell. 40 such diseases have already been reported, and the ratio or the percentage is not percentage, ratio is 1 to 8,000. Now every 8,000 individual, one person is suffering from such lysosomal disorder. अब इन डिसऑर्डर की जो है वो सवियरिटी जो है वो बेयरली डिटेक्टेबल से लेके वेरी सीवियर कंडीशंस भी हो सकती है। मींस अ पर्सन सफरिंग फ्रॉम lysosomal storage disorder may never realize that he is suffering from this disease, or in other cases that can be a fatal disease. कुछ डिजीजेस का मैं यहाँ पे हवाला दूंगा। आई सेल डिजीज है जैसे, फिर पॉम्पे डिजीज है, अब आई सेल डिजीज, अच्छा इससे पहले डिजीजेस के ऊपर बात करने से पहले देयर इज अ क्वेश्चन। हमने प्रोटीन्स के हवाले से हमेशा बात की है, देयर इज स्पेसिफिक अमीनो एसिड सीक्वेंस विच इज रिस्पॉन्सिबल to drive or to direct the protein to go at a specific place. Research has told us that the mannose 6 phosphate residues, आपको याद होगा हमने कल के लेक्चर में बात की थी कि प्रोटीन के साथ जो ग्लाइकोप्रोटीन कोर अटैच होती है उसमें हमारे पास मेनोज अटैच होता है साइड कोर के ऊपर. These mannoses are basically responsible to direct the proteins to the lysosomes. In other words, we can say that the proteins or lysosomal enzymes, they have an address written on them, and that address is in the form of mannose 6-phosphate, and that mannose 6-phosphate brings these enzymes to the lysosome. So, eye disease or eye cell disease is a very rare disease, and in this case, what happens that the lysosomes are full of undegraded material. The materials are present in the lysosomes, and when experimental studies were conducted using fibroblast cells, it was observed that material is accumulating in the lysosome. The enzyme is synthesized normally, but it is released in the cytoplasm or out of the cell. It is not going to go to the lysosome. So ultimately, where is the problem? The problem which was found was with the address, that is with the mannose residues of the core, which was not able to direct or which was not able to target the enzymes to go to the lysosome. Ultimately, you can say that there is a problem. There was no mutation, but there was an error in the address of these proteins, and that lead to development of eye cell disease. Then there is another disease which is Pompe disease. This happens because of absence of beta glucosidase enzyme. As a result of absence of beta-glucosidase enzyme, the glycogen starts accumulating in lysosome. And as you know, glycogen is a carbohydrate, is a polysaccharide, and when it accumulates in the lysosome, तो यहाँ पे फिर वो एक जो है वो osmotic disbalance create होता है, because glycogen is in the lysosome. It may need excessive amount of water. That water is derived from the cytoplasm. So this osmotic disbalance is going to create problem for the cytoplasm as well. And this beta glucosidase absence can be due to mutation in the gene or any other factor that this enzyme is not available. And the third disease is Tay-Sachs disease, which also results in the deficiency of due to the deficiency of enzyme beta N hexose aminidase A enzyme. और ये एंजाइम जो है that is required to degrade gangliosides. Now, if you can recall the ganglia or ganglioside, these are part of the nervous system. Nervous system में जो है GM2 is a major component in the brain cells, and if the enzyme is not present, the gangliosides they keep on accumulating in the blotted lysosome. और अगर ब्रेन सेल्स में वही अफेक्ट हो कि लाइसोसोम्स इंक्रीज हो जाए, साइटोप्लाज्मिक जो है कंपोजिशन डिस्टर्ब हो जाए, सेल्स are like full of the lysosomes, then dysfunctioning starts, and ultimately there is direct effect on the communication of the body with progressive mental disorder and motor retardation, as well as there will be effect on the skeletal cardiac muscles and respiratory, because skeletal muscle, cardiac muscle या फिर respiration के हवाले से बात करें, in all these three processes there is involved cellular communication. So if cell through nervous system, अगर nervous system जो है वो abnormally behave करेगा तो ये सारी की सारी जो है body की normal activities they are going to suffer. And for the Tay-Sachs disease, the ratio is 1 out of every 3,600 newborn, as reported in Europe, and this study was done on Jews. What are the therapeutic approaches in case of lysosomal disease. There is a disease which is Gaucher disease. This disease has been successfully treated by enzyme replacement therapy method. As the name indicates, that if you don't have an enzyme, or if you have an enzyme which is not functional properly, there is possibility that you can administer the enzyme by yourself to replace the defective enzyme and see if the disease is relieved or not. The patient is suffering, got some betterment or not. For Gaucher disease, there is an enzyme which is glucocerebrosidase enzyme. When this enzyme is not available, it causes the enlargement of spleen. And as you know, spleen is the site where the red blood cells are recycled. If spleen is not working well, then the person will suffer from anemia because of low number of red blood cells. Now, if we try to provide this glucocerebrosidase enzyme, then will there be any treatment or not? The two different attempts were made. In one attempt, what was done, that a normal human enzyme was given in the form of an injection in the bloodstream, but this was not successful. This therapy was not successful because blood circulation में जब ये enzyme liver तक पहुंचा तो liver took it as a drug which was administered from out of the body and retained that enzyme for further processing कह लीजिए या उसकी digestion कह लीजिए. फिर उसके बाद एक और जो अप्रोच यूज़ की गई वो अप्रोच ये थी कि मैक्रोफेजेस जो हैं उनको टारगेट किया गया इस एंजाइम के ज़रिए. बट टारगेट करने से पहले एंजाइम वाज प्यूरिफाइड not from the human blood. It was purified from the stem cells or the placental tissues. If you have some idea about placental tissues, large number of stem cells are found in the placental tissues along with other enzymes. So अगर वहाँ से आप एंजाइम को एक्सट्रैक्ट करें और using different glycosidases, you remove the terminal sugars on the enzyme oligosaccharide. Now I will recall yesterday's lecture again. This was a protein. यहाँ पे आपके पास एक बाइफर्केशन थी, मेनोज बने हुए थे और दोनों साइड चेन्स के ऊपर फिर ग्लूकोज और फिर सियालिक एसिड, ये जो है वो carbohydrates attached थे. Now what you did actually, you took this enzyme and you removed the terminal glucoses. You removed all the terminal carbohydrates, leaving behind only the mannose core. Now, this mannose can target any cell having the manosyl receptors. So, the idea was to remove the terminal carbohydrates, leaving behind a core with the mannose, and then inject it, and then it was injected to the patients. And this therapy remained successful because this enzyme, it traveled through the body and it went to those cells which has the receptor for the mannose. That was the macrophages, which took the enzyme by endocytosis and then granted some kind of therapeutic relief to the patients. So this is about enzyme replacement therapy and few diseases of lysosomal disorder. If you have any question, please ask.